So, analgesia is the relief from the perceptual experience of pain. And an analgesic or a painkiller is a drug that's used to achieve this sort of pain relief specifically. It differs from like an anesthetic drug, which actually can impact sort of all sensation, when administered. And that's sort of more like, adding like lidocaine topically to the gums, for example, during some sort of dental procedure where you get that complete sort of numbness. But analgesics are designed to sort of really go after the perception of pain, and pain often begins with detection of damaged tissue in the body, which is called nociception. And there are specialized neurons that are called nociceptors. These are specialized sensory neurons. And they actually, they look kind of like this. They're pseudo unipolar cells with a single cell body, cell body and a single pole coming off of it. That's in the dorsal root ganglion of the spinal nerves, or in the ganglia, for example, of the trigeminal nerve, which feeds the face, which basically brings sensory information from the face. And they have these endings, in the skin, for example. And these nociceptors will actually express receptors that can bind proteins that are released from damaged cells. So cells that get ripped or torn or cut or crushed, they spill out their contents, which are not supposed to be in the extracellular environment there. And these specialized nociceptors will, their receptor proteins in the membrane will bind and result in a depolarization. And this will send information back, to the central nervous system. And ultimately, it'll reach maps in the brain, particularly in areas like the insular lobe, and be experienced as, pain. So there's a lot of interest in drugs that can interfere with that process and sort of remove, the perceptual experience of pain. And so one of the behavioral assessments or techniques that is utilized to sort of see if a drug is having an impact on an animal's sense of pain, for example, is something called the tail flick assay. And this is used obviously with animals with a tail, like rodents. But basically the tail is sort of placed on something that can heat up, like an element that can heat. And then they'll raise the temperature. Sometimes they use a laser light, to basically, go onto the tail and slowly increase the heat that can ultimately at some stage do a little bit of injury. And as it rises, the animal will flick its tail away. So this is called the tail flick assay. And you look at the latency, like how many seconds it takes of, that heat on the tail, to generate the flick away, presumably because it hurts. It's unpleasant. And so if you give morphine, for example, to a rodent, it will have a much longer latency, many, many more seconds before it'll pull its tail away. And you can look at, for example, cannabinoids, other drugs that have impact over our perceptual experience of pain, and see how they, also influence, or act as analgesics, based on the tail flick assay and the amount of time it takes before the animal, responds to a injurious